So, proteins. By the way, proteins, they are the largest organic molecules, even larger than lipids. Yani the lipids, they are larger than carbohydrates. Proteins are larger than both. And they, again, chemically are formed of carbon, hydrogen, together with oxygen and nitrogen. If I have a protein only formed of these elements, so I call this a simple protein. But if I attach to it any other element like phosphorus, like sulfur, like magnesium, so this is not simple anymore. It is called conjugated protein. Okay, so either I have simple protein, CHON, CHON, or if I attach to it any other element, so it is a conjugated protein. Type. Proteins are formed of repeated units of amino acids, whatever the number is. But can I say that proteins now are polymers, ya Salman, or not? Proteins are formed of repeated units of amino acids. So can I say it's a polymer?
Yes.
Yes, of course. So protein is a polymer, and the amino acids, they are its monomers. But I cannot say polymer about lipids because they are formed of two different molecules, glycerol and fatty acids. But here they are. I have several amino acids. Yes, they can be different from each other in their kind, but they are all amino acids. So they are monomers that will form a polymer. So how many amino acids do we have? We know 20 amino acids only. So how do we use these amino acids? Do we have to use all of them to form a protein? No. Proteins, they differ in their types according to three factors concerning the amino acids: the kind of amino acids used, their number, and their sequence. How are they arranged? Now let me explain this. Suppose I have 3 amino acids. Let me call one of them A, the other amino acid is B, the other one is C. These are 3 different amino acids, 3 kinds. Their number, I'm going to use them in different numbers. For example, I'm going to use at the beginning A amino acid only. If I attached amino acid A to another amino acid A, it means of the same kind. Yani I used only 2 amino acids, the number and the kind. They are the same kind, only 2 of them. So I'm going to get a kind of protein. Let me say protein 1. If I used 3 amino acids of the same type, this will give me a different protein, protein number 2. Now let me introduce the BIBA. If I have amino acid A means similar to each other, and one is different, this will give me a different type of protein. Now let me change the sequence of these 3 amino acids. I'm going to put the B amino acid in the middle. This will change the protein. I'm going to get the B in the beginning. This will change the protein. Look at this. And I'm just using 2 amino acids only, and I still can get another probabilities for using. Suppose I put the B together, only 2, this will give me protein number 6. Suppose I took 3, will give me protein number 7. It means whenever I change the kind, I change the number, I change the sequence, I get a completely different protein. That's why I can never judge how many proteins are there. Yani I can say, what are the types of carbohydrates? I can say, what are the types of lipids? What are the numbers? What are they? But I can never say how many proteins can be formed. So I say that proteins are uncountable. I can never know the number of proteins that can be formed because I have 20 amino acids. I used only 2 of them here, and I got 7 different proteins, and still I didn't use them all. So according to the kind of the amino acid, according to the number I am using, according to how are they arranged like this, when I change where B is found, I can change the whole amino acid, the whole protein. So according to these 3 factors concerning the amino acids, I can change the whole protein. Type. Is that clear up till now? Or you want me to add something or to re-explain something?
Clear.
Okay. No questions, Salman?
No.
Okay.
No, no.
Right. Let's go now to this monomer. What is an amino acid? Let me explain it at the beginning and then I get you this picture. Amino acid. Amino acid is an organic acid. Remember what I said about the fatty acids. I told you that these are organic acids and they must contain which group? Who can remember? If you don't remember, it's okay. This is the first time you hear this. Huh? It must contain what? All organic acids must contain COOH. Okay? But how can I connect this to an amino acid? Because amino acids are organic acids, so they must contain carbon. And this carbon is the most important part in the structure of the amino acid. So this carbon, I said before that it is tetravalent. It means that I have to connect something around this carbon. So here I just put COOH. This is the carboxyl group. Or I can call it the acidic group, or the group that tells me that this is an acid. On the other side, I'm going to attach another group, NH2. This is called the amine group because it's called amino acid. Here, I will attach only hydrogen. Okay, let me write this a bit further. This is the amine group. So what about the 4th attachment? I'm going to just write R. So what is this R? Again R? Yes, it is a terminal group or a radical again. So what is this terminal group? In all amino acids, this part is constant, is found in all amino acids. If I change the R group, the whole amino acid will change. Now, what is this R group again? This is just a molecule. Whatever the molecule is, okay? So if I change this molecule, so the whole amino acid will change. But this part is stable. In all amino acids, it is found. So if I have an amino acid number 1, for example, so I put here R group number 1, a certain molecule. If I want to change the whole amino acid, this means I'm changing only the R group and I'm not changing this stable part. Okay? Let's go to examples. Now, I said that amino acids, they are formed like this: hydrogen, NH2, and R. I just said that this part is constant or is found in all the amino acids. Type. Let me give you an example for this R. Instead of R group, I will write here hydrogen. Can this be a group? Yes, it is. And this is the simplest amino acid. Do we have to know it? Yes, you have, because this is the simplest one. And of course, it is very simple because you have to memorize this part, and the R is just hydrogen. But what is this amino acid called? It is called glycine. Do I have to memorize it? Yes, because I'm going to use it later on, this glycine. Glycine is the kind of amino acid, or the simplest amino acid. Why simplest? Because the R group is just a hydrogen atom. Just a hydrogen atom. So this is very, very, very simple. But suppose I want to write another amino acid. So I'm going to write like this: carbon. Let me erase this بقى عشان أعمل لكم comparison كده between both of them. So I have here another amino acid. This is stable. I'm going to write it again. NH2. I will change the R only. Let me put another molecule. CH2 SH. This is different from H. So this is another amino acid. So the R group differed than this R group. So it gave me another amino acid. This one is called cysteine. Do we have to know it? No. No, you don't have to memorize it. Although it is very simple. But you have to memorize the structure of glycine because it has the simplest R group, which is only hydrogen. Type. I give you now example for two different amino acids. Why are they different, ya Habiba?
Because the R group is different.
Yes, because the R group is different. This R group can be as simple as this, or it can be a very large group. Okay, the R group differs from one amino acid to the other. And as you can see here, that this part is the same. We never touch it. Type. How can I make بقى protein from amino acid? I have to join the amino acids together because I just said that a protein is a polymer that is formed of amino acids, which are monomers. But how can I join them together, ya Salman? Like any other organic molecule, I want to join the monomers together. What should I do? What should I do in order to join the monomers together?
Untitled bond.
Okay, how can you do it? Of course, طبعا.
I don't know. What was that?
لا, you know. طب how can you join two monosaccharides together? Of course, you know. If you have two monosaccharides, how can you join them together to give a disaccharide?
Good.
You have to remove what? طب Habiba, we have to remove what?
Water.
Water, yes. I couldn't hear you, Salman. What did you say?
I forgot.
You forgot. Okay, never mind. But now you will not forget again. So whenever you want to join monomers together or even any molecule together like the triglycerides, you will remove water immediately. But let's remove water from 2 amino acids. Now and again, this is condensation, by the way. I have here, this is 1 amino acid, NH2. This is the R, let it be R1. And you don't have to memorize any R except that of glycine, which is hydrogen. And this is the COOH. I will put beside it the 2nd amino acid, hydrogen. But look, I'm going to write it exactly like this. Sorry. NH2. And here is the R, but number 2, because I will use 2 different amino acids. I want to remove a water molecule from them, from both of them. So I have to write this in a different way, exactly as I did before for the fatty acid, double bond O and OH. Why? Because I want to bring water molecule, components of water molecule beside each other. This is COOH. Now I want a hydrogen from here, so I'm going to write this in a different way. I'm going to write it like this. NH, and this is the other H. Is the NH2? Yes. And I will remove a water molecule from here. Water. Again, this is a condensation reaction, and it's going to form a kind of bond. Condensation. It's going to form a bond when I remove this molecule of water. So I will remove water, and I will see what is the bond that is going to be formed. Here is the bond. Yes, between the carbon and the nitrogen. And this is called, of course, peptide bond. So I have done a condensation reaction, and I removed a molecule of water out so I could attach the 2 amino acids together by a peptide bond. This is condensation. But what if I want to break it by Salman? What should I do? I want to break this bond. What should I do now? Yani I removed water in order to join them or to put the peptide bond. But I want to break them. Yes. So I'm going to return back this water molecule. So the bond is going to be broken. I will return the OH here and the hydrogen here once more. Okay, so I break it down. Right. I'm going to, of course, this is a hydrolysis reaction. When I break it down, it is a hydrolysis reaction. It means breaking down in the presence of water. Now, I'm going to revise with you again. Whenever I want to build up a great molecule or a macromolecule, why didn't I say a polymer? Because I'm including the lipids here. So I'm going to do 2 things: to remove water and to put a bond. And this reaction is called the condensation reaction. Now, what if I want to break down a large molecule or a macromolecule? I'm going to do the opposite. I will add water and I will break the bond, and this is called a hydrolysis reaction. Never forget that, okay? Never forget this. And by the way, most of these reactions, they need enzymes in order to speed up this reaction. And most of all our digestion, our digestive process all the reactions are hydrolysis reactions or breaking down reactions, because when we eat proteins, so we break them down in our digestive tract into amino acids. Whenever we eat lipids, they are broken down in our intestine into fatty acids and glycerol. Whenever we eat carbohydrates, they are broken down into their monomers in our digestive canal. So all digestion is a breaking down reactions of different macromolecules. Now, I'd like to show you this picture بقى to revise what I said. Now, as you can see here, that, of course, this is not good. I'm going to write it C, hydrogen, COOH, R, and NH2. Type. Should I put the hydrogen here and the R here? No, you can exchange them. Should I put this here and this here? No, you can exchange them. Yani it's not a must to put them, but to be more organized or to understand it better, you have to be fixed with a certain distribution of these molecules around the carbon atom. Now let's go here to this amino acid. Don't look at these, okay? This is the COOH, and this is the NH2, yes?
But what do they mean by inverted?
By?
Inverted.
Inverted means that, okay, it means that they put it opposite wise.
Yeah, why?
Type. Here, as you can see, that the R1 was here and the R2 was here. It's just inverted, but it doesn't have any meaning to us. As I just told you, I can put the hydrogen here or here. It doesn't matter. Okay, because they wrote it in two different ways.
Yeah, so it's correct if I did it in the same direction.
Yes, of course. As I told you before, that this has no significance. There's no importance to us. Either you write it like this or like this. They wrote it here in order to show you that you can write this or that. But it has no importance to us. So here, I'm going to remove one water molecule, means condensation, to form this peptide bond. But please don't draw the peptide bond like this. Why that? Because this is a covalent bond. Covalent bond, this is not the symbol. They are just drawing it like this to show you that this is something that is done here. If I want to break it down, I will just add water to return them back to the two amino acids, and this is a hydrolysis reaction. Type. If I made a protein that is formed only of two amino acids that are joined together the same way by condensation reaction forming a peptide bond. So if I have a protein that is formed of two amino acids only, I call this protein a dipeptide. One of my students, once upon a time, she told me, does this mean there are two peptide bonds? No, no, no. Dipeptide means a protein formed of two amino acids. طب how many peptide bonds, ya Habiba, must be found between them? Between the two amino acids, how many peptide bonds? I have two amino acids and I joined them together. One, yes, very good. One peptide bond only. This means that this di has nothing to do with the bond. It means that this protein is formed of two amino acids. This di stands for the number of the amino acids. Now, what if more than two amino acids? So, it is a polypeptide. There is no tripeptide, no. It is immediately polypeptide. If 2 amino acids, then the protein is called dipeptide. If more than 2, then it is a polypeptide. And because I told you that proteins can reach huge length of amino acids, we call it a polypeptide chain. Okay, because it looks like a chain. طيب, who can tell me if the chain is very long, what will happen to the molecule? If the chain becomes very long, what will happen to the molecule? Who remind me of this? Because I forgot it. If I have a very long chain, I mentioned this about the amylose, about the lipids, the hydrocarbon chain. When it becomes very long, what is it going to do?
Energy is used to break it.
No. If I have a very long chain like this, so it either
It should coil
or fold. I drew this for the fatty acid, and I mentioned this in amylose. Remember? Remember, Salman? Yes or no?
No, but now I remember.
Type. Whenever you find a very long chain, it's going to coil or to fold. Why? In order to shorten this great length and be put in a small space. I mentioned this in polysaccharides when I told you, yes, it's going to be compact. Very good. So because it has to be included in a small area, so it has to be compact like this, either to coil or to fold. And the same happens with protein, because the protein molecules are very long. So they are going either to coil or to fold. And this leads me to a part here that I have to explain, which is called the protein levels of structure. Now, if I have protein that is just formed of amino acids that are joined together by peptide bonds, just like that. Just amino acids with peptide bonds, whatever the length is. I call this structure the primary structure. Primary means the simplest, or number one, yani, the simplest structure. Just amino acids that are joined together like this. So joined, of course, by peptide bonds, and they just form a chain. So this is called the primary structure. What if this chain becomes longer? So it is either going to coil like this or to fold like this.
Secondary structure.
Yes, the secondary structure. So this is called a secondary structure. But what's the difference between both? We have, if it is coiling, we call it the alpha helix. If it is folded, we call it the beta pleated sheet. And by the way, the coiling never uncoils, and this pleat never unpleat. But how can they be joined like this to prevent their uncoiling or returning back to a primary structure? Who can think with me? Does anything bind these coils or these pleats together? Who can tell me? And they can never uncoil or unpleat. What does this mean? This means that there is a bond holding the coils like this. And I want now the name of this bond, please. What is the name of the bond that has this structure? Huh? What's the name of the bond? This was mentioned in the first session. What's the name of the bond that has this symbol? Dotted line. No.
No. Hydrogen bond.
Hydrogen bond, very good. Hydrogen bond. So there are hydrogen bonds between these coils in order to prevent their separation to return back to this shape. No, they do not return because of the presence of this hydrogen bond. But what about the pleated sheet? Again, there are hydrogen bonds like this holding this shape in place. Sometimes we have several pleated sheets beside each other like this. And they are again joined together by hydrogen bonds in order to prevent their separation. So here I have only peptide bond. What about the secondary structure? I want you to think with me. The secondary structure, does it have peptide bonds as well or not? إيه رأيكم? Salman, please, I want you to participate with us. Try to think with me. وحبيبة كمان, you have to think. Is there a peptide bond in the secondary structure or not? Yes or no? If you don't know, I will lead you to the answer now.
I think so. I'm not sure.
Okay. Salman, what's your opinion?
They are joined together by peptide.
Yes, of course. Yes, طبعا يا حبيبة. You said I think so. ده طبعا. Why بقى?
Because it's protein.
Yes,
right?
it's a protein. But it became very long chain. That's why it coiled. It means that the primary structure has only a peptide bond between the amino acids. Secondary structure has two bonds, peptide and hydrogen bonds. Okay? Now, what if this structure became more complicated? They are going to fold more and more and more. The folding here, let me erase this and draw it here because I need the space to show you the difference. And here are the hydrogen bonds binding these together and these together. Now, what if the secondary structure became more complex, or it started, in spite of the alpha helix and the beta pleated shapes, they started to coil back. So they are going to give me a more complicated structure that looks like this. Suppose this is the beta pleated, it's going to form like this. Something very much complicated. This structure, or if I want to draw it as the alpha, so the alpha is going to look like this.
Tangled.
Excuse me?
It looks tangled.
Yes, but it has a name, a level at the beginning. The level here I have primary, secondary. This is
Tertiary.
Yes, tertiary. But why we call it tertiary? Because this is the level number 3 according to complexity. So we say tertiary structure. And if you looked at a tertiary structure protein, you will find that it is in a 3D shape. It looks like a ball, ya jama'a. It looks like the globe. You know what's the globe? It is the earth, rounded, spherical. So we say that the tertiary protein is globular. If I want to describe what's the shape of the tertiary, I say it's globular. It looks like a globe. It is in 3D shape. But the secondary, I say it's fibrous because it's not folded much like this. Fibrous means that it looks like a fiber. It is long like this. Yes, it is coiled or pleated or helix or beta pleated sheet, but it's long. So it forms fibers. Fibers means something looking like this. Yes, here it is made pleated or coiled, but it is still long. It's not globular like this, looking like a sphere. Type. I mentioned now that there are hydrogen bonds that attach the parts together to prevent their unfolding or uncoiling. But what about these? How can they remain in these shapes? Who can tell me what is present here to prevent their uncoiling like this? Who can tell me? There must be something that holds the shape like this.
Covalent bond?
Excuse me?
Covalent bond?
لا, I'd rather to say bonds only. Bonds. Because here I'm going to introduce other bonds. So let me revise with you. Here I have peptide bonds only. Here I have peptide and hydrogen bonds. But here I'm going to mention other types of bonds. Okay? So I have to erase this and tell you what are the types of bonds in the tertiary structure. In the tertiary structure, I have the original peptide bond. I have, again, hydrogen bonds. Why بقى? Because I said that the tertiary structure is folding imposed on a secondary structure. طب ما secondary أصلاً فيها hydrogen bonds. So, hydrogen bonds are still there. But there are other bonds بقى. A bond called sulfur-sulfur bond. Remember, I mentioned this bond in this first session we had, and I told you that I'm going to introduce it later. But what's the meaning of sulfur-sulfur bond? Means that the bond is between what, يا جماعة?
Two sulfur atoms.
Yes, excellent, both of you. طيب, انتوا عارفين إن الsulfur atoms, when I draw, طبعا كل ده between amino acids, okay? If I have amino acid like this, and I said this is constant in all amino acids, and the R group is a kind of molecule that changes from one amino acid to the other. But where is the sulfur in this part in which the bond is going to be formed? Is there sulfur here? No. Then where is the sulfur? Excellent.
The R.
Yes, excellent. This means that if the R group contains sulfur, and I have two amino acids in this shape, suppose an amino acid here contains sulfur, another amino acid here contains sulfur, so the sulfur-sulfur bond will form between them. You can imagine this. If I have sulfur in an R group here and an R group there, so immediately a bond will form between them, as if it's forming a bridge between the amino acids. So that's why we can call this the sulfur bridge. I can say that this bond is sulfur-sulfur bond, or you can say that it is a sulfur bridge. There is another bond. This bond is an ionic bond. Now remember, what is an ionic bond, يا جماعة? Who can remind me? When I mentioned between, attraction, very good. Between what, ya Salman?
Metal and non-metal.
Okay, it means between which charges?
Positive and negative.
Positive and negative. Excellent. طيب حبيبة, what's the difference between the ionic and the hydrogen bonds? This is again between positive and negative. What's the difference between them?
An hydrogen bond slightly negative.
Excellent, bravo. Bravo.
But an ionic, it is strong
very
attraction.
good. طب انتوا عارفين بقى, there is something that I didn't mention in water. I told you that hydrogen bonds are weak or strong?
Weak.
Weak, yes. But if I have many hydrogen bonds, they are going to be strong. That's why the water molecules are held together tightly. Okay, that's why I cannot break the bonds except when I heat 200 degrees Celsius. So hydrogen bonds, yes, if I have one, it is weak. But if I have many, they are strong. في التجمع قوة. Okay, here in the tertiary structure, I have all these bonds, and all these bonds, they hold the tertiary structure in its globular form, like this. Can never be unfolded except I can have here a tertiary structure. It is coiled much like this, giving me a 3D shape, and it has all these bonds, so it does not separate from each other, except by changing the pH or by heating. Exactly like what, ya jama'a? What are the kinds of molecules that are damaged by different pHs or by heating?
Enzymes.
Enzymes, excellent. طب انتوا عارفين ان the enzymes, they are tertiary structured proteins? That's why we say that they denature by heating and pH. طب why بقى؟ الsulfur-sulfur bond والionic bond, they are found between the R groups. يبقى الانزيم الR groups بتاعته, they are separated from each other or they become unjoined when we change the pH or the heating, because they are held together actually by sulfur-sulfur bonds or ionic bonds, which are denatured if we change the pH or the temperature. So this is the third type of the protein level of structure type. Let me go to the fourth kind بقى. The fourth kind ده سهل. It is called quaternary. Quattro means four. Quaternary structure. Quaternary structure, there are four tertiary molecules. And I'm going to show you now an example. Molecules. So if I have three tertiary structures like this, polypeptide chains طبعا, but they are taking this form. This is number 3 and number 4. So this is called the quaternary structure, and the best example here is hemoglobin, which I'm going to show you now. So the best example for a tertiary structure are the enzymes. The best example for the quaternary structure is the hemoglobin molecule. It means that in hemoglobin, again, I have the same bonds that were found in the tertiary. Why? Because a quaternary structure is formed of 4 molecules of tertiary structured proteins. But let us revise together back. And you're going to remind me. Primary structure. What are the type of bond found here, ya Habiba?
Peptide bond.
Peptide bond only. طيب secondary structure يا سلمان, which type of bonds are found here? We have the secondary.
Peptide and
Excellent, excellent, bravo, bravo. Peptide and hydrogen bonds. طيب حبيبة, the tertiary structure. معلش, this is the hardest بقى, معلش. So we have peptide,
hydrogen,
very good,
sulfur,
excellent, excellent, sulfur, and ionic. Okay, طب سلمان, can you tell me the quaternary? What are the types of bonds there?
peptide, hydrogen, sulfur, and ionic.
excellent. برافو عليكم. برافو عليكم. طب let me بقى show you picture for these. Now here, this is a tertiary structure. But let me begin by the primary بقى الأول بقى والحاجات دي يعني. Oh, where are they? Here are they. But this is a very poor drawing. It's found in the book. This is a primary structure. Look how the amino acids are joined as if they are lining up. And of course, there is a peptide bond between them. These are the two shapes of the secondary structure. This is an alpha helix, this is a beta pleated. But of course, I have to draw here the hydrogen bonds in between as dotted lines, like this and like this. This is a tertiary structure, and this is a quaternary, but of course it must have another one. I don't know how in the book they drew this. Look here at this picture. Here, this is tertiary. But why does it look like this? Because I said that the tertiary structure is folding, don't forget this statement please, imposed on a secondary structure. And here are they. You can see here, this is a beta pleated sheet, where these are the hydrogen bonds holding them. This is an alpha helix, and these are the hydrogen bonds forming them. And this is a primary structure as well. Here I have a disulfide bond or a sulfur-sulfur bond. It means that here is an amino acid that contains sulfur and another one that contains sulfur. So this is a disulfide bond. Of course, this picture does not show, well, it's found here. No, it does not show the ionic. I'm going to show you. I hope, بس alhamdulillah, I added them. Look at this. طبعا pictures دول, they are not from Edexcel book, they are from Cambridge book, but they are very clear. This is a tertiary structure. Look at the coil. And look at the folding. يبقى في هنا a fold, or the pleated sheet I mean. This is a folding imposed on the secondary structure. Let's look back at all the bonds. Look here. I have hydrogen and oxygen. There is a hydrogen bond here, eh? Okay? Right. This is an amino acid containing sulfur and another amino acid that contains sulfur. This is a disulfide bond, or a sulfur-sulfur bond, or a sulfur bridge. Now, look here. I have here NH2. This is nitrogen with the O negative. So I have here the NH2, sorry, it is positive, and the O negative, so this is an ionic bond. Okay? And look at this one. This is a quaternary, and this is the hemoglobin molecule. This is one tertiary, another one, another one, another one. So this is called quaternary. And if this is folded much more than this, so I can see the tertiary structure clearly. Now, I told you before that the alpha pleated or the beta pleated or the alpha helix, they act like fibers. So I call them the fibrous proteins. So if I want to mention their structure according to their function, I will say that I have two types of proteins, fibrous and globular, from their shapes. Fibrous means that I have either alpha helix like this, I will not draw the hydrogen bonds, or beta pleated sheets. So they look like fibers, they are not globular, they are not looking like a sphere, but here the globular must look like this, something like this. Okay, so the fibrous, they are formed of secondary levels. But here they are either tertiary
or
or yes, or quaternary. And because of this structure, they have a function. يعني, as I told you before, there must come a question. Relate the structure to its function. So how can I relate this structure?
enzymes. Oh no,
هنا الانزيمز yes. So, yes of course. طيب الفايبرس يا جماعة دايما بتبقى structural protein. But what does this mean? It means that it gives structure to the organism. And the best example here that we are going to learn is the collagen. You know that the collagen is found under the skin and it gives a structure to the skin. Yani if somebody is at your age, your skin is healthy, it is stretched because there are several factors, and collagen is one of these factors that are present in your body that allows the skin to have a certain structure. So I call collagen a structural protein, and it is fibrous, as we are going to learn. Also, collagen does not only and is not only found in the skin, it is found also in the blood vessels, giving a structure to the blood vessels, giving them a certain shape. Also, collagen is found in bones. Do you know that collagen is very strong? When we become older in age, this collagen becomes weaker. That's why the skin becomes wrinkled. And that's why some people, they go to the doctor to take Botox and things like that, to inject collagen under the skin in order to return the skin to its healthy shape. But here, the globular, they have a certain function. That's why we call all the globular proteins functional. Like what, yes, Salman? You just said it. Enzymes, they have a certain function. They are globular. Antibodies, they have a certain function. They are globular. Hemoglobin, it has a certain function that it is globular. So whenever, and of course the proteins that are found in the cell membrane, they are having a function for the passage of particles and water and things like that. So globular, they have a function and we call them functional proteins. But the fibrous proteins, they give structure to the organism. That's why they are structural proteins. But let me start by the fibrous proteins. Number one, fibrous proteins. Fibrous proteins, they are mainly secondary. Yani it's very rare to find a fibrous protein tertiary, of a tertiary structure. Very, very rare. They are all, yani either they are not tertiary at all, only secondary, or sometimes yani we find a little amount of globular protein, but you don't have to know this. Here we have polypeptide chains that are found parallel to each other like this. Or, of course, the coiled ones, again, they are parallel to each other. Several polypeptide chains, they are found together and they are parallel together. And of course, they are held together by hydrogen bonds to prevent their separation and to keep their structure. All the fibrous proteins, they have a certain property in that they are extremely tough, very strong, and they are all insoluble in water. All of them. Whenever I find something that is insoluble in water, I say that it forms a colloid with water, as I told you before when I was explaining water. But see here, this is very important property for the fibrous proteins. Like, where are the fibrous proteins found? I just said them. They are found in all the connective tissues. Let me erase that in order to write where are they found. They are found in connective tissue. Now, what is this connective tissue? It's a tissue that connects all the parts of the body together. And this is, by the way, the mostly found tissue in our body as animal body, because these kinds of proteins, of course, they are animal in nature. But also, they are found in the tendons. And I'm going to explain now what's the meaning of a tendon. They are found in bones. They are found in, together with muscles, like the muscles that form the wall of the blood vessels. They are found under the skin. They are found in the hair, or they form the keratin, our fibrous protein keratin, that is found in hair and in our nails. Okay, what else? Okay, not only for humans, they are found in the horns of cattle, also in the feathers of the bird. All these contain keratin, and all of them, they are fibrous proteins. Now, the best example, as I just said, is the collagen. And this is the one we are going, inshallah, to learn next session.